I am pleased to be hosting Dr. Thomas Quiroz Monnens and Dr. Alice Boulanger to discuss the recent PLOS ONE Lab Protocol paper entitled A Large-Scale Bacterial Attraction Assay, a new quantitative bacterial migration assay suitable for genetic screens, and the associated detailed protocol in protocols.io. Alice is a molecular microbiologist with a strong focus on pathogenic interactions and the regulation of virulence traits. Her expertise spans both plant and human pathogens, where she has gained broad experience in host pathogen dynamics. During her PhD with Professor Matthew Orlott at the LIPME INRAE, she uncovered how Xanthomonas campestris utilizes N-acetylglucosamine during infections, expanding our understanding of bacterial strategies. In his postdoctoral fellowship at the NIH with Dr. Deborah Hinton, she explored the molecular mechanisms governing virulence gene activation in Bordetella pertussis. Later, at CNRS in Marseille, she investigated prophage maintenance in Salmonella typhimurium. Since 2015, she has been an assistant professor at Paul Sabatier University in France, leading projects focused on the genetics of Xanthomonas citnus in plants. Thomas Quiroz Monnens is a plant pathologist with a focus on pathogen genomics. He obtained his bachelor's and master's degrees in plant sciences and plant biotechnology in Wageningen University in the Netherlands. His studies focused specifically on plant pathology. He very recently obtained his PhD under supervision of Alice Boulanger at the LIPME INRAE in Toulouse. During his PhD, Thomas studied the genetics of virulence and fitness of the plant pathogenic bacteria Xanthomonas genus, with a focus on Xanthomonas campestris, Pathobar campestris. Thank you so much, both of you, for being here.
Thank you for the nice invitation, Gabriel.
So to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, so in order to explain these things, I think it's important that I first introduce a little bit about bacterial chemotaxis and about our work that we are doing that has motivated us to publish this protocol. So bacterial chemotaxis is the directed motility of a bacteria in response to chemical gradients. And bacteria make use of chemotaxis in order to navigate their environment. There are environmental bacteria that use chemotaxis to find food. And there are pathogenic bacteria that make use of chemotaxis to find openings in their hosts. So chemotaxis has been studied for many decades, and the molecular mechanisms are quite well understood in some model bacteria like E. coli. And I cannot explain all the ins and outs of chemotaxis signaling, but it boils down to the fact that there are specialized chemoreceptors that we call MCPs that can bind chemicals called chemoeffectors. And once they are bound, there will be a signal transduction that includes a variety of genes, different steps of phosphorylation and methylation that eventually control the directional rotation of the flagella and thereby give directionality to the movement of the bacteria. But not all bacteria are created equally. And we know that what we find in E. coli does not necessarily translate to what we find in other bacteria. And others can encode more complex signaling systems that can involve additional genes and different kinds of regulators. So in our team, we study a plant pathogenic bacterium, Xanthomonas campestris, which causes disease on cruciferous plants like cauliflower. And in order to study how this bacteria causes disease, we make use of random barcoded transposon insertion site sequencing, or RBTNSEQ for short. In RBTNSEQ, we start with a wild-type population of bacteria that we mutate by the insertion of a transposon. And this leads to a heterogeneous mutant population. And with these kind of populations, we perform a variety of different screens. We can, for example, inoculate our population in vitro or in planta. And mutants in the population that are mutated in genes that are important to overcome this kind of pressures in vitro and in planta will become depleted from the population. If we then sequence the bacterial DNA of the different populations and compare these with each other, we can identify genes that are important for the bacteria to overcome these evolutionary pressures. So we were also interested in chemotaxis and the role of chemotaxis during the infection of Xanthomonas. And we envisioned that we could use our RBTNSEQ library to identify genes in Xanthomonas that are involved in chemotaxis. We thought that we could expose our library to a chemical gradient and physically separate mutants that are able to sense and move to these gradients from those that cannot. And it would be very nice to do this in planta, but the problem is that if you want to study chemotaxis in planta, you need to use very low inoculum densities and low numbers of bacteria. And for TNSEQ screens, we need high numbers of bacteria. So natural systems did not suffice. And then we thought that we could maybe use some sort of artificial system to separate these kind of mutants within our population. And that gave yield to the large-scale bacterial attraction assay. So it comes down to a Nalgene filtration beaker. And in the middle of the beaker, we place a 1.2 micron filter, which is a pore size that is just big enough for XCC to swim through, or for Xanthomonas to swim through. We then fill our system with a buffer, add the bacteria to the bottom compartment of the system, add a chemoattractant to the top, and over time, the chemoattractant will diffuse to the bottom, prompting bacteria to migrate to the top if they are attracted by this particular component. So that is the essence of the large-scale bacterial attraction assay. It is a cheap and accessible method that we can use to collect high numbers of chemotactically attracted bacteria. In the PLOS ONE publication, we have deeply characterized this system, making use of Xanthomonas campestris and also Shewanella oatniensis. We have used this particular system to perform forward genetic screens in Xanthomonas to identify genes that are involved in chemotaxis signaling. But that does not mean that it has to end with Xanthomonas, because there are many other mutant libraries available in different types of bacteria. And this system could be used by others that are working on these kind of libraries to screen for genes involved in bacterial chemotaxis.
Thank you, Thomas, for the very thorough explanation. And as part of the motivation for you guys to develop this assay, you mentioned that doing it in planta would have been ideal, but you need a large quantity of bacteria. So how about other pre-existing in vitro screen methods? Why they didn't work for you?
So with a previous PhD student, we tried in collaboration with other group using a really well-known tool as a capillary system, where you have really small capillary that are filled with chemoattractant, and you just you can have many different capillary that will just touch the surface of a bacterial suspension. And in fact, it's working well. Even with Xanthomonas, we have positive control. So it was working, but the problem was the amount of bacteria, even if we use many capilla, collected was not enough to get enough bacteria DNA. So the preparation of DNA was really difficult in this condition, and to sequence after it was not giving nice results. So we stopped there, and Thomas arrived, and he's a super creative, smart person, and he was really interested by find another way to perform this chemotaxis assay with our library, and he tried many things, and he found this system, and it was working really great. So we were super happy.
The protocol, it's so elegant. It is very simple, seems very affordable. I don't know if there are any critical steps. Like, is there anything that the user has to be pretty careful with?
Yeah, I think in the technical part there are some steps that you have to take into consideration, and one of the things is the formation of air bubbles in your system, because you have the bottom compartment which is filled with a liquid and the top compartment which is also filled, and in the middle there's this filter. But if there is air in the bottom of your system, it can block the water flow between the bottom and the top compartment, and then you cannot have migration from bacteria that move from the bottom to the top. So it is very important that there is no air in your system.
And I'm very curious now to know, what else did you try, Thomas? Like, did you immediately thought about this, this protocol, or was there anything else that you tried that didn't work, didn't result in a final protocol?
So the system now consists of the big beaker, and this is working. But before, we tried many things. We tried with Falcon tubes and gluing filters to Falcon tubes. This didn't work. We have tried to do similar things with filters with syringes. So we had syringes that had a filter attached to them. But all these systems, they were too small. We could not collect enough bacteria. So yeah, in the end, we settled with this, and this was the one that worked best.
And again, the protocol is elegant, is simple. I wonder whether for those genetic screens that you have performed, have you had to do any further optimization?
Yeah, we have tried different kind of concentrations for attractants to be sure that it was working well. We have tried a few different type of buffers in the system as well to see which kind of buffers were working best. So for Xanthomonas campestris, we used a 1 millimolar MgCl2 solution as a buffer, which was working fine. But when we were trying to use the system with another bacteria, with the Shewonella, we saw that with this buffer, there was no migration to the top compartment. And we were very surprised by that, because they were more or less the same size. We had we used mallet as an attractant, which is a known attractant for Shewonella. So we were very surprised to see that we didn't observe any migration. And in the end, we figured that the bacteria, in fact, they get stuck to the filter, the Shewonella. So we found in literature that there were people, when they were doing these kind of or other type of experiments with Shewonella, to stop them from sticking, they would add a little bit of tween, which is a detergent, a surfactant, to the system, and that stopped the bacteria from sticking. So we also use tween in our system, and this allowed the bacteria to move and not to stick to the filter.
And so that's a point to consider if people want to transfer the protocol to other bacteria.
Yeah, I think if anyone want to use it with its favorite bacterium, there will be some step of engineer. They will have to engineer the system to their own bacterium. So you need really a good positive control that you know that it work well. If you start from nothing, it would be a bit difficult to check that everything is working well. And you need a bit of background. And you maybe also need to adjust the spore size of the filter depending on the size of the bacterium that you're looking at also.
Thank you. I would like to change gears a little bit now and discuss about the motivations of your team to have the protocol peer-reviewed as a standalone methodological paper.
Well, when we discussed at different meeting and congress with other collaborators or other phytopathologists, a lot of them were really, really interested by the way we were doing this. So we thought that the best way to be useful would be to publish a protocol and to peer review.
And how did the feedback that you received from the peer reviewers help you shape the final version of the protocol?
You want to answer that?
Yeah. So the peer review was quite tough, but was fair also, because we are not chemotaxis experts. So I believe that in the initial version of the protocol, we had missed some controls that were not so obvious to us, but that were more obvious to people that were specialized in the field. And we believe that the reviewers were specialized in chemotaxis. So they gave us a lot of valuable comments that we could use to improve the protocol, that we used to perform additional experiments to further substantiate our claims. And they also helped us to rewrite a few things that we were maybe not phrasing in the best way, I think, yes. So this was very valuable to us.
And my final question might be a little bit speculative at this point because the paper is relatively young. It was published early in the summer. But how do you see both the protocol and the paper impacting your careers at different stages? An established scientist and a young fellow that just graduated. Congratulations.
Thank you. Well, for me, this was actually a very important step in my career, because for my doctoral school, this required that we have one first author publication in order to graduate. And this was the first paper that I published as a first author in my PhD. So for me, this was the paper that allowed me to graduate, which was very nice, and took a bit of pressure of the journey, let's say. But I also really hope that others that are having similar libraries as us will see this protocol and be like, okay, this is a simple experiment, it is accessible, we have these kind of tools laying around, so we can also perform these kind of screens for genes involved in chemotaxis. And perhaps, like, when others will also do this, we can identify new genes and regulators that are involved in chemotaxis that are maybe epistatic to the known chemotaxis signaling genes.
Yeah, and we were also already contacted by other groups. One group in particular in Spain, Madrid, that is a specialist in chemotaxi and Pseudomonas chemotaxi. And before it was published, but they want to use it and maybe collaborate. So we told them that we will publish the protocol, we will give it to them, and if they want to have more input and also videos about how you do it, we can share. So it's nice.
So I don't have any more questions. I don't know if you want to add anything that we haven't discussed so far.
I think the protocol IO is really a useful tool. It's really nice to have something really nicely shaped with this tool. So I think this was a plus also for the protocol. And thank you very much for the opportunity to talk about this protocol.
And elaborating a little bit on your response about the features of protocols.io, and what is really nice is that once the protocol is published, if you find that protocol potentially useful for your own bacteria, as you said, they might need some engineering to do, but in one click with the fork, they can transfer all that information and just tweak those steps that they need to improve to make it work in the condition.
Yeah, it's really nice.
Hmm.
I think it's really, really nice. We didn't know about it before, so now we will look at it much more.
Well, thank you so much for your time.
Thank you, Gabriel. Have a good day.
Thanks.